You are quiet now. The day has gone soft around the edges, and the world feels folded in shadow. Every cell, every organ, every hair on your arm knows the alphabet of life. Not one you learned in school, not one you can speak. And yet, they form the deepest script you've ever known. Deed another code that builds you. But it is there, 4 letters whispered through time, A, T, C, and G. A double helix coiled like a sleeping vine, turning inward in endless repetition. Billions of rungs, each one a pair of letters, holding hands across a molecular ladder. You lie still, your breath moving like the slow rhythm of tides. In this gentle stillness, you begin to notice something smaller, quieter, the hum beneath the hum, the silent music inside you. Your eyes, your lungs, your skin, it all began with this folded message. A message copied and carried, generation after generation, like a lullaby passed down without sound. Even as you sleep, the instructions unfold, telling your body how to heal, how to replace, how to endure. And sometimes they become challenges, illnesses, conditions, struggles. But even the flaws carry story. They remind us this is not a machine, it is a living language. Your body, warm and resting, holds an ancient language. Their cells carried this same code, slightly different, slightly reshaped by time. Their lives wrote small edits into your story, and now you carry those edits forward, like punctuation marks in a sentence still unfolding. Adenine, thymine, cytosine, guanine, these are not words. They are molecules, not thoughts, but instructions. As cells divide, the message copies itself, checking for errors, adjusting, persisting. A library that keeps renewing itself, 1 letter at a time. Billions of years of biology distilled into the spiral of your cells, not digital, not mechanical, but biological. A slow whisper in the dark. To change a letter in the middle of a sentence. That is CRISPR, but not yet. Tonight is not about the tools. From the first flicker of life in ancient oceans to this very breath you retaking now, it is all part of the same story. You imagine it now, spiraling softly in the dark, not for its perfection, but for its persistence. It does not boast, it does not erase. It remembers. A to T, C to G, always together, always in pattern. You imagine strands of DNA unfurling, like ribbons of silk, each one curled tight around coils of memory. In laboratories, scientists unwind these strands and read their sequences, letter by letter, like decoding a hidden script. Like poetry you've never read, but somehow always carried. You feel it in the mystery of why you are you, and not someone else. You breathe slowly and feel the quiet miracle of it. This code is in every cell of you. It told the cell how to grow, how to divide, how to become more. It didn't explain it, simply guided, quietly, reliably, a map drawn in molecules. You were once a single cell, and inside that cell, this alphabet was already written. Tonight you walk along that map not with feet, but with thought. But the story isn't theirs, it's yours. Because every blink of your eyes, every beat of your heart is built on this code. This code does not shout. It does not change quickly. It is slow, intentional, faithful. Sometimes it makes mistakes as small typos in the script, and sometimes those typos become traits, a freckle, a curl, a difference. You think of the people before you, your ancestors. This isn't just your body. It is a book begun long before you were born. You rest in that thought, letting the weight of time settle gently on your chest. And tonight you listen. You listen not with ears, but with awe. Because something inside you knows that this alphabet, so quiet, is sacred. You imagine the DNA writing itself over and over again, and now scientists have learned to read it, to edit it. Tonight is about the text. You lie still, floating gently in the awareness that your very being was once a line of code. A, T, C, G, the song your cells still sing. You may never see your own DNA, but you feel it in the steadiness of your heart, in the shape of your thoughts, in the softness of your skin. You are not a machine. You are a message. And tomorrow, the question will change from what is written to what could be rewritten. But for now, rest. Let the letters hum. Let your cells remember. Let the code keep turning. You are a sentence still being written. You lie still, the hush of night surrounding you like a soft blanket, but inside, a story stirs quiet, microscopic, ancient. A story that begins in the smallest corners of life, where survival isn't loud but clever, where the tiniest beings, bacteria, face the vast threat of invisible invaders. Long before humans spoke of genes or cures or codes, bacteria lived in the deep and the dark, surrounded by dangers. Among those dangers were viruses called phages, molecular pirates that slipped into cells, hijacked their machinery, and left only destruction behind. But the bacteria were not defenseless. Somewhere in their simplicity, they developed something extraordinary, a way to remember past invaders, to store tiny pieces of viral DNA like warning signs, a molecular memory. And when those same viruses returned, the bacteria were ready. They used a system called CRISPR, an acronym for something long and technical, but tonight you don't need the full name. What matters is what it means. A tool born from defense, honed by evolution, perfected in silence. It searches for matching code, and when it finds it, it cuts. CRISPR is not a weapon in the usual sense. It is more like a pair of scissors made of enzymes and intention, cleanly, precisely, without fuss. You imagine it now, floating through the tiny architecture of a bacterial cell. The same scissors that sliced viral DNA are now reshaping the future of medicine, agriculture, even identity, of how something so small could ripple into something so large. A bacterial defense turned into a human hope. A protein named Cas9 drifts gently, scanning each strand of DNA like a librarian checking shelves, revealed, recognized. You feel the weight of that transition, not heavy, but meaningful, a pause between breaths. Who watched a protein cut a strand of DNA with such grace that they whispered, "What else could it do?" They saw the precision, the elegance, the simplicity, and they asked, could we borrow it? Could we guide those scissors, not just to defend, but to heal? When it finds a piece that matches its memory, the fragment from an invading virus, it makes its move. A small cut, a tiny act of survival, and that would have been the end of the story. That in the unseen corners of the world, life has always been clever, always been resilient, always been writing its own quiet story of survival. Except that humans noticed. In labs filled with glass and light, scientists watched the system work. The answer still unfolding is yes. Tonight, you rest in that moment before transformation, before CRISPR left the world of microbes and entered the human story. Because the same mechanism that once protected a bacterium is now being used to edit human genes. You float in the quiet awe of it. It feels almost magical, but it is not magic. It is science at its most humble, watching, listening, learning from nature not to conquer it, but to collaborate. CRISPR was not invented. It was discovered. And now, in careful hands, it becomes more than memory. It becomes possibility. As you drift deeper into rest, you imagine the first scientists who saw this. What diseases could it stop? What broken genes could it mend? What burdens could it ease? But for all the questions, there is also caution. Because tools, once discovered, must be held with care. Tonight, you are not holding anything. You are simply noticing. And now, for the first time, we are part of that story, not just as readers, but as editors. You breathe slowly, thinking of that humble microbe and its silent scissors. Not dangerous, not loud, just ready. Ready to defend, ready to help, ready to change everything. You lie still beneath the ceiling of thoughts, soft and slow. The world around you fades into quiet, and in that hush, you feel it. Not fear, not excitement, but a quiet responsibility. A kind of stillness that comes when you hold something fragile and powerful and new. Tonight, you imagine holding scissors, but not made of steel. To change even one letter is to edit that story and to write a new one, and cut with the delicacy of a whisper. Then a choice, repair the break, insert a new phrase, or silence the faulty line. Not heavy or cold. These scissors are invisible, made of enzymes and intention. CRISPR, once the guardian of bacteria, now rests in your grasp, and with it comes a question: where will you cut? No louder than a protein drifting in a dish, no faster than a cell dividing, but behind each use, a thought: should we? You picture DNA still and humming, a double helix glowing like moonlit ribbon. Each strand is a sentence, a story, a lineage. The tool is precise, astonishingly so. Guided by a molecule designed to recognize a sequence, CRISPR can slip into the genome, find a match. It is editing at the scale of dreams. It's not about miracles, it's about precision, it's about care. It reflects who we are, what we value, what we dare, what we hope to fix. The gene for a disease, we want to change it. But what about the gene for height or memory, or something we don't yet understand? Because when you hold scissors this fine, you must move slowly. One child saved from an inherited illness. One crop made stronger without chemicals. One community given hope through a molecular correction. When the paper is as delicate as life itself, each snip matters. And yet it is not science fiction, it is now. In labs around the world, CRISPR has been used to treat diseases, to modify crops, to explore how life itself might be shaped. They rest in the minds of those who think carefully, who choose slowly, who feel the hum of responsibility beneath the hum of possibility. You float in that space between can and should. It's easy to feel awe, but the real story is quieter. You breathe deeper. CRISPR is not just a tool. it's a mirror. Where is the line between healing and enhancing, between fixing and redesigning? You're not here to solve it tonight. You're here to rest in the presence of that question, to let it settle around you like mist, because technology does not shape the future. We do, and we are learning how to use scissors made of light and thought. The promise is real, but so is the challenge. The temptation to reach too far, to edit for perfection, to forget that nature's script includes pauses, imperfections, and poetry. You are part of that script, not flawless, not finished, but full of meaning. And CRISPR, like all powerful things, reminds you your choices shape not just your story, but the stories of those yet to come. So tonight you lie still, the scissors are quiet now. You are not here to cut, you are here to imagine. To feel the weight of a tool so precise it can change a letter in a word billions of years old. To wonder what kind of authors we will be, not just in science, but in compassion, in care, in courage. CRISPR may cut with enzymes, but what it shapes will be written with thought. You are weightless now, drifting through possibility that feels both wondrous and wary. Beneath your fingertips, the story of a future child unfolds not in books, but in base pairs. It begins not with breath, but with a code, where the future stands quietly and asks, "What will you make of me?" 3 billion letters, folded inside each cell. And for the first time, we hold the means to edit it before life begins. This is the place where science meets ethics. Germline editing, that quiet, profound shift in a fertilized egg's DNA means that what we change today could echo through generations. It's not medicine for one. It's a message passed forward, carried in blood and breath and bone. CRISPR can remove a mutation. It can add resistance. It can even alter traits we once believed unchangeable. But when the child is born, they will never know what was altered or what was left untouched. They hum beneath every conversation about designer traits, about enhancement, about fairness and consent. And so you float now not through a lab, but through a question. Can we change the script of life without changing what it means to be human? You imagine a newborn sleeping in silence, a body shaped by choices it never made, a life that begins with a gentle silence because somewhere someone chose to prevent a pain, to rewrite a word, to correct a gene. Will that difference carry weight? Will it divide? Or will it disappear into the normal messiness of being alive? Is that love? Is it design? Is it both? These questions are not loud. They do not shout, but they persist. You drift through that hum. You think of their children and their children's children, each carrying a note in the symphony of inheritance. The science is breathtaking. We have already seen immune diseases undone, rare disorders halted. The way your breath feels longer here, the way your thoughts soften under starlight. The door is open, but it is a heavy door, and the room beyond is delicate. They sit with these questions in rooms filled with quiet light, not to rush, but to reflect. You picture a future schoolyard where some children carry edits and others do not. You don't have answers. No one does not yet. What we do have is pause, a global conversation, a gathering of ethicists, doctors, parents, philosophers. Because when you change a blueprint before the building begins, every beam, every shadow, every echo is shaped by that choice. And tonight, you two are sitting in that room, not with fear, not with judgment, just with openness, with wonder, with responsibility. You think of the children who will laugh, run, fall, grow, never knowing how their cells were touched by science. You do not need to decide tonight. You only need to feel the weight of this quiet, of this moment. Because editing tomorrow's children isn't about power. It's about care, and care moves slowly. So you rest here in the hush of not yet, in the silence before the answer, in the reverence before the edit. The child is still asleep. The code is still whole. And the future waits not for certainty, but for wisdom. You sit now in the soft quiet of a memory, not your own, but one held in DNA, a silence that spans generations. A family shaped by a single mutation, a condition passed gently, painfully through blood. And here you are, not at the end of that story, but at a new beginning. With it, we have begun to reach into the deepest places of the body, not to erase identity, but to offer relief. CRISPR has become the scalpel of the genome. Sharp, swift, astonishing, rare names, rare burdens, but real lives, real pain. Real possibility now for healing sickle cell anemia, Huntington's disease, Tay-Sachs. You imagine a child whose bones no longer ache, an adult whose neurons no longer fray. These are not miracles, they are molecules. Here, researchers hold pipettes like pens. They write in the language of proteins. Each droplet is a sentence, each sequence a poem. You walk into a small lab where the light is gentle and the air smells like clean metal and blooming hope. They are not trying to make better people. They are trying to make better days. Because healing is not about changing who someone is. It's about lifting what weighs them down. You watch a patient receive edited cells, tiny couriers of care. These cells carry no promises, but they carry intent. CRISPR is not here to erase scars. It's here to prevent wounds that never needed to be. Inside them, a gene has been tuned, not deleted, not rewritten from scratch, but softened, corrected, aligned to listening. And as those cells take root, something shifts. With patients who understand their own genes better than any textbook ever could. Machines sleep, the genomes rest. What if the edit removes more than pain? What if it silences a trait that carried strength, resilience, even joy? It's about helping them live fully without fear. It's a hand outstretched, offering help, not change for its own sake, but change that respects. Not just in the patient's blood, but in their sense of tomorrow. You feel your own cells, busy and quiet, pulsing with life. This is what it means to heal without erasing. To honor the path someone has walked, while easing the road ahead. To say, you are not broken, but if you are hurting, we can help. You think of scars, those quiet reminders that something happened, and you survived. And yet, with every step forward, there must be pause. The line between healing and altering is fine as a strand of DNA. So we return again and again to intention. Healing must begin not with scissors, but with stories. With families who know the cost of inherited suffering, their voices guide the edits. Their truths inform the thresholds. You breathe in, slow and full. Around you, the lab fades into night. In the stillness, one truth glows. Medicine is not about making people perfect. And CRISPR, when used with humility, is not a force for erasure. You lie back beneath the soft hum of stars. Healing, you realize, is not a finish line. It's a gentle unfolding. And CRISPR is not here to erase your story. It's here to help you write the next line. You step into a garden where the air smells like dusk and dew, where the ground breathes gently beneath your feet. But here, nothing grows quite as it once did. Corn that no longer invites insects. The leaves look the same, but something invisible pulses within their stems. Edited code. Precision grown in Petri dishes, now planted under stars. Wheat that resists drought. Rice that survives floods. A tool of efficiency, of resilience, but also of philosophy. Because changing crops means changing systems. Changing systems means changing lives. A gene once responsible for spoilage has been snipped, trimmed like a thread in a sweater. Now they last longer, waste less. In countries where deficiency blinds children, these grains may give back sight. Environmentalists worry about wild species, cross-pollination, unforeseen consequences. One edit, one nutrient, one ripple of change. You crouch beside a row of tomatoes. Their skin is smooth, their growth strong, but this garden is not a miracle. It is a decision. CRISPR in agriculture isn't flashy. It hums, silent and steady, inside every seed. Fields that survive climate's lash. Communities that plant not for profit, but for people. It's warm, forgiving. Life still grows here, edited or not. Just a tool held carefully by those who sow with wisdom. You wander through rows of golden rice, engineered to contain more vitamin A, but not all are convinced. Will these changes travel beyond the field? Are we disturbing a balance we don't fully understand? You hear the debates in the wind. Should we edit what we eat? Farmers raise concerns about patents, about dependence on corporations, and yet hunger waits for no debate. You feel the tension beneath your feet, roots pulling both ways, toward tradition, toward innovation. In this garden, the goal is not dominance, it's resilience. To feed without stripping, to grow without draining, to adapt without erasing the past. You imagine a future harvest, not just abundant, but ethical. Food that nourishes bodies and respects ecosystems. And in that vision, CRISPR is a quiet partner, not a savior, not a master. You reach down and run your fingers through the soil. This is the garden that grows differently, not perfectly, not permanently, but with intention. Close your eyes and imagine your earliest self, not your childhood, not your first memory, but the moment before all memory. A cluster of cells, dividing, silent, already carrying the entire story of you. Inside those cells is a sequence. A long, elegant sentence written in the alphabet of life. A code so intricate, so ancient, it hums with something more than logic. It hums with longing. But as we peel back the layers of DNA, we also peel back layers of self. Then you float further into genes that affect memory, mood, risk. Into regions where science still whispers, unsure. And somewhere in that spiral of adenine and cytosine, of guanine and thymine, we begin to wonder, is this where the soul lives? This chapter is not about science alone. It is about something deeper, the quiet question that trails every discovery. Are we just code? It cannot measure a laugh or predict a sacrifice. CRISPR can change genes. It can reshape bodies. It can lift burdens. You drift through the sequence. You see the gene that decides your eye color, your ability to taste bitterness, the shape of your earlobes. You begin to feel a tenderness, not for the precision of the code, but for its poetry. This is not a blueprint. It is a ballad, a slow song passed from mother to child, father to son, woven with both history and hope, all shaped by genes, by life, by chance. You think of a person whose DNA carries the mark of a condition, but who has also grown strong, kind, radiant because of it. Not to overwrite the story, but to add a footnote of kindness. Below you, your cells whisper, busy with the business of being. CRISPR is powerful, yes, but it is not the soul. It does not hold love or fear or the ache of a farewell. And yet, it touches the place where these things begin. You think of identity, gender, personality, memory, none written in stone. Would editing that gene remove the trait or the triumph? You see now, the soul may not live in the sequence, but the sequence is part of the soul's house. To edit with care is to touch something sacred, not because we are playing gods, but because we are walking beside the mystery of what it means to be human. You remember a phrase from a scientist's notebook? We do not edit to perfect, we edit to understand. And maybe that is the point. Not to conquer the genome, but to learn its language more gently. You breathe in deeply. Above you, the stars wheel silently. And in between, your thoughts float not in fear, not in certainty, but in wonder. You are more than your DNA, but your DNA is still part of your song. And in that song is a single shining note. I am here. I am human. I am whole. Imagine standing at the mouth of a vast canyon. You whisper a thought, a question, and wait. What comes back is not just your voice, but an echo shaped by stone and wind and time. This is the nature of ethics. It is not immediate. It is not always loud. But it is what gives shape to science. It is the voice that returns when we ask not how, but should. But the echo that follows each edit, the consequence, the perception, the legacy, cannot be edited out. CRISPR has changed what is possible. We can now edit genes with breathtaking precision. Scientists, doctors, ethicists, patients, and parents sit in a quiet circle. There are no pipettes, no microscopes. Only stories, questions, doubt, and care. Tonight, you listen to that echo. You picture a meeting room warm with soft light. They speak not just about what genes do, but what people feel, what communities fear, what culture remembers. Because ethics lives where science meets society. One voice wonders, will genetic edits widen inequality? Will those with resources edit more and those without be left behind? It stretches across cultures, across belief systems, across generations. Another asks, if we edit for intelligence or beauty or strength, who decides what traits are worthy? Who gets left out of that definition? To resist the pull of speed in favor of depth, the room does not rush to answer. It holds space. That too is part of ethical progress, to slow down. And so we weave these threads slowly, carefully, like DNA itself, and in many hearts, the question still rests unanswered. You float above this conversation, not to escape it, but to see its scale. Ethics is not a wall, it is a net. In the stillness, you imagine a future shaped by consensus. Not by the loudest voice, but by the clearest heart. A future where CRISPR is used with dignity, justice, and humility. What one society deems acceptable, another may fear. What one generation believes is right, another may regret. In some countries, laws already guide what CRISPR can and cannot do. In others, the framework is still forming. You feel the echo in policy, in protest, in poems. Because every time we change the code of life, we also change the story of being human. But ethics is not a brake on science, it is a compass. It does not stop us, it steers us. And the echo it offers is this: what we do with power defines who we become. You close your eyes. That future begins not in the lab, but in reflection. And tonight, you are part of that reflection. Not with answers, but with presence. Not with certainty, but with care. The ethics are in the echo. And your listening is part of the sound. You sit beside a stream, watching water weave its way through roots and stone. It bends, it adjusts, it rewrites its own path without ever stopping. And you realize this is nature's way. Adaptation is not invention, it is revision. Life learns not by knowing, but by trying again and again across millennia. Radiation, error, time, each mutation a chance, most failed. A few survived, and those few wrote the story forward. Long before any lab, nature had its own editing tools. You think of the finch with a slightly broader beak, better suited for the seeds of a changing land. Of the bacteria that, by accident, resist a drug. Of the human child born with a rare gene that helps them thrive where others weaken. Within each, a copying machine hums not flawless, but faithful. Errors slip in, most are caught. Some are passed on. Every child is a variation. Every organism, a remix. These are not miracles, they are edits. But we do it with a new awareness that every gene exists within a system, every edit inside an ecology. Each changing what traits matter, what lives, what leaves. CRISPR, then, is not new. It is human hands learning to write in a script older than language. You watch cells divide. And in this, there is no shame. Nature does not seek perfection. It seeks survival, balance, beauty born from unpredictability. CRISPR is our attempt to join that rhythm, not to master it, but to dance with it. We borrow nature's scalpel and try to guide what nature once left to chance. You look around and see adaptation not as accident, but as intention shaped by time. The giraffe's neck, the orchid's trap, the eye's light-sensitive dance. You picture an ice age, a virus, a volcanic ash cloud, each an editor in its own right. And so we step gently because the soil we edit rests atop billions of years of silent edits before us. We are not the first editors, we are just the first to know we are editing. And with that knowledge comes humility. You rise from the stream, feeling its chill in your bones. Nature's edits are everywhere. And CRISPR, wielded with wisdom, becomes not an intrusion, but a continuation. A dialogue, not a declaration. And in that dialogue, we ask not just how to change, but how to listen to what has already changed us. You stand now in a room without walls. It hums softly, like the inside of a seashell. All around you are strands of DNA, gently turning, folding, whispering futures you cannot yet see. It curves, loops, tucks itself into spirals. Each fold changes how it is read, what is hidden, what is revealed. And just like thought, just like history, the structure of what we carry matters as much as the content. The code is not flat, not only to replace or remove, but to reshape the way the genome is held, wrapped, lived. A therapy delivered not by surgery, but by a drop of mist into the lungs. They laugh, they play, they never wonder what could have been. Their world has changed not with fanfare, but with quiet shifts. Less disease, more resilience. A map redrawn gently. A mutation corrected before a symptom begins. A future parent, editing not for perfection, but for peace. CRISPR touches that fold. You reach out in your mind and feel the shape of possibility. These folds carry more than biology. They carry intention. Because what we choose to edit reflects not just what we know, but what we hope for. You imagine a child born in a decade already carrying edits made with care, but there is also risk. Precision that once seemed fiction now hums in refrigerators and notebooks. Forget that every human carries a unique, intricate origami of self. In folding new futures, we may crease the old ones, erase diversity, flatten the unexpected. Your body, this astonishing library of folds, breathes with you. Every cell, a sentence. Every heartbeat, a rhythm of what was and what might be. So we hold this power with grace. We fold with slow hands, with eyes wide open. We ask, will this edit help or narrow? Will it include or divide? Will it leave space for surprise? Because the future is not just a destination, it's a responsibility. And the genome is not a blueprint, it's a conversation. They open to possibility, a child saved, a harvest secured, a disease forgotten. In labs around the world, researchers now guide edits with increasing elegance, single-base changes, epigenetic nudges. But the beauty is not in the edit alone, it's in the why. You step back and see the folds expand. They do not open to certainty. Each fold is a fingerprint. And CRISPR is not the end of the story, it is the next line. You breathe in, and here, at the end of this chapter, you pause. Not to decide, but to wonder. Because inside every fold, the future is already forming. And the way you hold it matters.